Question one before we move on to the next chapter here. So the last thing to really talk about here is just taking a look at really how are these different macromolecules broken down and a little bit about their absorption. Again, don't want to get super crazy in depth on this one, but again, it's just understanding that each of these macromolecules are our building blocks of most of our molecules, and because of that, we need to be able to break them down into their basic subunits and really how is that happening. So carbohydrates, again, you can get up to starches, which are polysaccharides, but these are made up of a lot of these different disaccharides and monosaccharides that you've heard of before. So table sugar, for instance, is sucrose, which is two sugars worked together, and in most classes we've talked about glucose. So what we need is we need ways to start breaking this down. We know a little bit already that the oral cavity, we started with salivary amylase to start the breakdown of that. The other spot we see this is in the small intestines. Really, salivary amylase we talked a little bit about already. It's time to taking those starches, breaking them down into glucose molecules. Happens a little bit in the mouth. Supposedly if you chew on like a unsalted saltine cracker, which why would you ever want to do that? But if you do that for a little bit, supposedly it will get sweet. I've seen this once or twice when I've tried it. What happens though is once we swallow this down, that little bit of breakdown that started to happen is going to get inactivated, and really we're not going to have any other chemical digestion taking place in the stomach of these carbohydrates. It's really once we get to the small intestine, we release pancreatic amylase. So this is part of that pancreatic milieu of juices that it's kicking out there, and that is going to again start getting after these polysaccharides, breaking them down into their smaller units here, get down into these individual glucose molecules. There is also some specific ones that we see that are released by the intestinal wall. So brush border enzymes, these are the ones that the epithelium is releasing. So you can see some things like maltase, glucoamylase, dextrinase, lactase for those people who aren't lactose intolerant, can take this and break that lactose down into glucose and galactose. Again, all these ones here, again, the main thing getting at, if we're going to get these broken down, we have to have the enzymes to do it. And not surprisingly, between the pancreas and the intestines, we have what we need, with the exception of people with lactose and sensitivity issues, to be able to break down all these kind of moderately large starches all the way down to some of these smaller disaccharides, break them into their basic subunits. They're going to be absorbed across the small intestine. A lot of this is going to be either transport or diffusion. It's going to move its way in there. Transported up to the liver. A lot of these things are going to be modified through gluconeogenesis to glucose for the body to use. And if we need it, we're going to take and make glycogen, convert excess calories, excess sugars into fats if need be. Cellulose we can break down. We don't have the enzymes to do that. That ends up being part of what we excrete out. So you can kind of see here is these different enzymes of the pancreas and what they're doing along with the brush border enzymes, taking that polysaccharide, breaking it down into its basic subunits. And that's really what we're doing with these different enzymes here. You see amylase is really getting after the starch. Then you get these other ones, those brush border enzymes are really going to go after and clip those into those simple sugars. And again, you can see what some of these other ones would do. Again, I don't think I'm going to specifically ask you what sucrase versus lactase does, but I'm just letting you see it here. Proteins, similar idea here. We have all these amino acids. We need to have we have these large polypeptides. We need to break them down into their basic subunits. Again, certain enzymes are going to go after certain sequences of amino acids. Most of these are released as proenzymes, being activated. What this really thing is here is a lot of proteins within our cells. We don't want to start digesting our own cells, so that's what we have going on with these ones. Again, the main breakdown starts in the stomach with pepsinogen getting activated to pepsin. It is activated by that low pH. Some of that pepsin can activate other pepsinogen. And again, once we do this a little bit here, that chemicals, HCl is also denaturing some of the proteins in there a little bit, making them easier to get at. Once we get to the pancreas, we're going to see that there's a few other ones here. We have what is called trypsinogen, chymotrypsinogen, and procarboxypeptidase. Again, all these OGen on the end of it, those are both inactive forms. Again, each of these is going after slightly different amino acid sequences to be able to clip this. And the procarboxypeptidase, it's also going after a certain sequence, certain part of those amino acids. We do have some enzymes in the small intestine that activate this. So enteropeptidase is what's going to take trypsinogen and convert it to trypsin. Again, all these things, especially with the peptidases, that we activate them once they're in with what we've ingested. So trypsin can activate more trypsin from trypsinogen. Chymotrypsinogen gets activated to chymotrypsin. Procarboxypeptidase to carboxypeptidase. And again, all these pepsin, trypsin, chymotrypsin are going to break specific amino acid sequences. Carboxypeptidase goes and does that carboxyl end to the next amino acid, clips that bond. And then once we get down here, we do have also dipeptidase, which will finally clip these two amino acids from one another, and we get all those free amino acids. Again, through diffusion, osmosis, active transport, are going to get taken into the small intestines, go up to the liver. They can be converted. They can be, other stuff can be done with them here. But again, we're going to use these ones for the building blocks of making all of our proteins. And again, this just kind of shows you all these different ones and how they're clipping. Lipids, again, a little bit different with lipids, a little harder to get at. We have the bile that's going to break these apart a little bit. We have a few lipases that get activated as they get down here. Triglycerides, we break them down into glycerol and the 3 fatty acid tails. We need enzymes to do that. Cholesterol, we don't break it down to absorb it. We just absorb cholesterol. Lingual lipase, like I said, released in the mouth, activated once it reaches the stomach. Gastric lipase also made there. It's going to do a little bit of that fat digestion, so about 30% of it, taking those triglycerides and breaking them down. The biggest enzyme probably activating this is going to be pancreatic lipase. It is released once we get into the intestines from the pancreas and its little mix of juices that it's releasing. This takes triglycerides, converts them down to monoglycerides and fatty acids. Because we have the bile in this one, this is a much more effective one and is doing a lot bigger percent of that fat breakdown because of taking that emulsification step. Those big fat globs getting turned into little fat globs, giving the enzymes more to work on. And again, it is those bile salts going after this that really take those big fat drops, turn them into little fat drops. And we do recover a lot of that as we work our way through. Lipids are kind of interesting. They're kind of taken into these simple columnar cells. What's interesting about these ones is they are taken into the lacteals. They form something called chylomicrons that are released into these lacteals through exocytosis, those little vesicle merging with that and dumping its contents out. And they're going to get into lymphatic capillaries and then enter the bloodstream up where the lymphatics dump back in, and then we'll go to the liver and other tissues. So they don't go the same pathway as most of the rest of this. And you can kind of see it right here showing you kind of those chylomicrons getting moved through here and then being put into the lacteal. And again, a little bit different than most of the other nutrients we're talking about. Last thing here, nucleic acids. So anything that we ingest that has DNA and RNA, we can break these down to their basic subunits. So we have deoxyribonuclease, so that breaks down DNA, and ribonuclease, both enzymes from the pancreas that are going to clip between the bases of these DNA molecules. There's also phosphatases and nucleosidases that are going to clip the nitrogen bases from the sugars, clip the phosphates off. And again, what this gets us to is our basic subunits, which make a nucleic acid, which is a nitrogenous base, a sugar, and a phosphate. We can then absorb all that stuff, and when we need to, put it all back together to do DNA synthesis, other stuff along those lines. And again, just showing you the basic subunits of this. We clip these into all 3 of their own little parts here.